Hi, my name is Isabel Toghramadjian, and my project is optimizing photoperiod to improve drought tolerance in Arabidopsis thaliana. As climate change continues to escalate, we're going to have to adapt our agricultural methods to ensure that there will be enough resources to sustain our population, especially as there are changing and limited growth conditions and resources. It's been found that modification of the pseudo-response regulator gene in Arabidopsis thaliana, which regulates the circadian clock, increased cold, salinity, and drought stress tolerance, as well as biomass. And though extensive research has been done on changes in drought tolerance in plants prompted by direct genetic modification of the circadian clock, little research has been done regarding environmentally induced changes. So in my study, I sought to explore how changes in the photoperiod of a growth environment would alter the response to drought in Arabidopsis thaliana. I hypothesized that plants with longer hours of daylight would have a significantly greater decrease in both their mass and number of leaves when subjected to drought conditions than plants with shorter hours of daylight. So I programmed two different Conviron growth chambers with a night setting and a day setting, and I set one of them to have 12 hours of night, 12 hours of day, and the other to have 14 hours of day, 10 hours of night. And for each of those chambers, I prepared two different trays with 20 pots each, and then in each of those pots, I put three Arabidopsis seeds. And essentially, I did everything I could to try to get as many seeds to germinate as possible, and then I placed them into the chambers. So for the first 25 days after germination, I watered each tray once a week by pouring 500 milliliters of water into the base of the tray, um, with the idea that then the pots would absorb the water through their drainage holes. And then after those 25 days, I stopped watering half of the plants, so one tray per chamber, in order to simulate the drought conditions, and the other tray continued to receive water as a control group. Additionally, after that kind of drought breaking point, I changed my watering methods to also put 20 milliliters of water directly into each pot because I wanted to ensure that the plants would survive, so I'd actually have something to measure. 24 days after the drought, a total of 49 days after germination, I stopped my trials by breaking the stems of the plant at the surface of the soil. At that point, I counted the number of leaves that each plant had and then air dried each specimen at room temperature for 10 days so that I could then measure the dry biomass. The images here, I have the progression of the plants in the 1410 photo period. So on the top row are the drought plants and the bottom row are the watered plants. And you can see that the drought plants essentially died after I stopped watering them, and then the others continued to flourish. So for my results, first here I have the graph of the effect of that photo period and water conditions had on the number of leaves. So on the x-axis, I have all my different groups, so separated by water and drought, and then the different colors represent the different photo periods. And you can see that the watered plants had more leaves than the drought plants, and then especially in the water group, but um, in the watered group, excuse me, the 1410 plants had more leaves than the 1212 plants, although in the drought group, they're pretty comparable in terms of leaves. And then I ran a two-way ANOVA to see what impact the interaction between the independent variables of water and photoperiod had on the number of leaves. And for that p-value, I got less than 1%. Here, same general idea, except the dependent variable is now mass instead of leaf number. And again, you see that the watered plants, unsurprisingly, had a higher mass than the drought plants, and then the watered 1410 plants had more than the watered 1212 plants. And once again, the drought plants are pretty comparable between the different photo periods. I ran another two-way ANOVA here, and for that p-value, I got 0.126. Then finally here, I just have the percent change in mass and number of leaves between the water and drought conditions for each of my photo periods. So you can see in each case, the 1410 photo period had a greater decrease in the dependent variable than the 1212 plant, indicating that it was more affected by the drought. So though my goal was to evaluate the role that the interaction between the independent variables played. There was a few noteworthy individual relationships between each of the independent and dependent variables. So the plants that were watered, unsurprisingly, performed significantly better than their drought counterparts for each of the dependent variables and photoperiods, with p-values very close to zero, and this relationship was expected. The drought plants died soon after I stopped watering them, and the water plants continued to grow, so it makes sense that they had greater mass and more leaves. The plants that had 14 hours of daylight had significantly more leaves than those with only 12 hours, with the p-value of less than 1%. And because the increased daylight allowed more photosynthesis to take place, it's very unsurprising that the plants experienced more growth. In terms of the interaction, there was a significant relationship observed between the interaction of photoperiod and water conditions and the number of leaves on the plants, as I showed in figure 2. Although there was not a significant relationship observed with the interaction um, of photoperiod and water on the mass of the plants. So much more research should be done to determine whether this relationship is actually significant or if it was observed in the study as a result of flawed methodology. In terms of ideas to improve my study, first of all, there need to be many more trials involving a wider range of photoperiods. I didn't have the time or space in my chambers to test more than two, but it's really impossible to truly understand the nature that the relationship between photoperiod and drought play on a plant's growth without having a wider range of data. Additionally, more nuanced drought conditions should be considered. In my study, my drought condition was simply no water at all. And actually, I think it would have been better for the drought plants to still receive some water so that they could stay alive and maybe continue growth, but still less than the control group. And then that would actually allow the comparison of the growth of the plants in different conditions rather than just comparing living plants to dead plants, which is what my experiment ended up being. Finally, I think the duration of the trial should be extended because in my experiment, only the plants that had 14 hours of daylight and water um, actually ended up flowering. And I think that if there had been more time, some of the other groups could have flowered as well, and then that would have opened up other variables to consider, like hypocotyl length or flowering time. So here are my references, and thank you.